Hello student, welcome to the Diversity in the Living World NEET 2027. So we are going to discuss this second chapter now, that is called as biological classification. So first, when we are discussing biological classification, first scientist comes to the mind, and this is the one of the MCQ also, the Aristotle. Aristotle gave the earlier scientific classification. Hmm. So Aristotle gave earliest scientific classification. One of the MCQs asked, you can write Aristotle. Okay. Next, how he classified animals and plants. So simply for plants, he used morphological characters, simple morphological character which are visible. So one is growth habit. Growth habit means how much the tree is growing, whether it grows like a tree or it grows like a smaller shrub and even smaller herb. That is called as growth habit. Okay. Next is the color of the plants, leaves, bark, and everyone. And finally, number and shape of leaves. Number and shape of leaves. So Aristotle used all these morphological characters and gave us three categories of plants. One is trees, another one is shrub, another one is herb. Okay, so pretty simple, pretty clear idea. So Aristotle, earliest scientific classification, then used morphology characters such as these and give you categories such as these. Next is animals. So what is happening with animals? That animals could be anaema and could be enaema. So everybody knows anemia. Anemia is a what we call lack of blood. It's anemic conditions. Okay, so here animals without blood are called as anaema. Always remember, ana means lack, less of it. Okay, so anaema, animals without blood. Most of the insect, yes, most of the insect, water insects and all that, they don't have RBCs. Okay, next, enaima. Enaima is animals with red blood cells. Okay, so pretty simple classification. If red, enaima; if not red, anaema. And again, in terms of plant, again just trees, shrubs, and herbs. And this classification depends on what? Growth habit, color, and number and shape of leaves. So this topic is done. Very simple. Aristotle's earliest scientific classification. Next. Next, we are going with the more scientific and more proper classification given by Carolus Linnaeus. Okay, so Carolus Linnaeus is also called as father of classic uh, classification and all. So his name was actually Carl von Linné. He was a Dutch scientist. Later, he Latinized his name into Carolus Linnaeus. This is not given somewhere in NCERT. This is just for say knowledge. Nobody will ask that. Okay. Next. So his criteria was simple. If the cell wall is present, put that organism into plantae. If the cell wall is absent, put that organism into animalia. He did not go for any other criteria, complex criteria. It's very simple, and at that time it was revolutionary and it works very well for that time. But it created major drawbacks also. So cell wall are present in plants, bacteria, fungi. So all these were grouped under kingdom planting. So that's why some fungi have those names, dermatophyta. Phyta means what? Plant. Dermato means skin. So skin infectious fungi, one of those, that's why the name is like that. So kingdom planting. It is kingdom planting. Next is when cell wall is absent. Next is what? When cell wall is absent, protozoa, vertebrates, and invertebrates. Protozoa, vertebrates, and invertebrates. These were considered animals. This protozoa, single-celled organism, does not have cell wall. It can be considered as animal under two kingdom classification system. Okay, two kingdom classification system. So if they ask you what is the criteria, cell wall presence or absence, it does not, they are not He has not gone with the chemical composition, structure of cell wall, nothing, no complexity, simple. If there is a cell wall, yes, it is a plant. If there is not a cell wall, yes, it is a animal. So very simple, and this oversimplification has created some drawbacks also. So two kingdom systems drawback when we discuss, it will be like this. First, no distinction between prokaryotes and eukaryotes. See, eukaryotes with nucleus, prokaryotes without nucleus, they are placed together in plantae and also in animalia. Unicellular and multicellular. Same thing is happening. Unicellular bacteria or amoeba or protists like things are put together with multicellular. Next, one of the most important thing um, drawback in this classification is this photosynthetic algae is put together with non-photosynthetic fungi. That is also a problem because one is autotroph and another one is heterotroph. Next, the unicellular bacteria. Now pay attention here. Unicellular bacteria are grouped in plant. This unicellular bacteria are grouped with plant, not in plant, with plants. Okay, so that was a problem. And again, these dinoflagellates, diatoms, these were and protozoan, these resemble but kept in different kingdom. See, these diatom dinoflagellates, look at this diatom and these protozoans, they resemble in which way? They are saying resemble, but in which way? So they resemble being unicellular eukaryote. These are all unicellular eukaryote, but they are placed in a different kingdom under this only cell wall present, absent criteria. So pay attention to this. Third one is a fungi. Third one is a fungi. Now what is happening in fungi? Fungi is put in plant kingdom. Fungi is heterotrophic, while plant kingdom is majorly and mostly autotrophic. Fungi does not have chlorophyll and its cell wall is made up of chitin. You can call it chitin also, but it is called as chitin. And this chitin is non-cellulosic. So fungi heterotrophic, does not have chlorophyll, and cell wall composition is different. It contains chitin, not cellulose, as in majorly present in plants. So what is happening? Different because the classification criteria is so simple, presence or absence of cell wall, many people are clumped together which are not actually related to each other. Next. And fifth one, generally they will ask question based on this, is Chlamydomonas and Spiraea. Both are algae, both are green algae, but they are put together in algae in plants. But these are again unicellular and multicellular example. So this is what is happening. If you create a very simple classification, it can create so much problems because these members, I'm going to highlight these members don't belong to their particular so pay attention here. So if you go with the plantae, the members do not belong here are bacteria and fungi, bacteria and fungi. Okay. Okay, so as we were talking about kingdom, two kingdom system, and the problem with the simple criteria, that is whether the cell wall is present or absent. So this creates due to oversimplicity, this is creating problems. So we have placed bacteria and fungi in plantae, which they do not belong, and protozoa in kingdom animalia, also they don't belong. Okay, so this is the drawback of two kingdom classification given by Carlos Linnaeus. Next. So third, in that, third kingdom was added by Haeckel. Okay, remember, third kingdom was added by Haeckel. So the kingdom was Protista, and Protista is for unicellular eukaryotes. Very important addition. It resolved many of the unicellular and multicellular problem. Okay, so what problem they have solved? they have separated unicellular eukaryotes into protista. Very simple. Okay. Just one kingdom removed most of the problems. And they are going to be mostly aquatic. This you have to remember, because single cell can be either parasitic, or if it is going to be free living, it needs water for its metabolism and all the cellular process. Okay. Next came the fourth one, Copeland, and he gave Monera, very important. So all the prokaryotes, there was a problem of prokaryote in eukaryote. All the prokaryotes he separated in one kingdom. He called it Monera. So now eubacteria, major group of prokaryotes are eubacteria and archibacteria. Eubacteria are true bacteria, archibacteria are ancient bacteria. There are some difference, but both lack nucleus. In eubacteria, who comes cyanobacteria, mycoplasma, these organisms come. So this kingdom was added by Copeland. This kingdom was added by Copeland. Okay. So this is where major addition of what new kingdoms took place. Now we are coming to very important part, that is R.H. Whittaker's five kingdom system. Okay, so five kingdom classification system given by R.H. Whittaker, which is considered as a standard. Okay, it is still mostly used. So he gave particular criteria. So now what is the criteria here? It's cell structure. Cell structure means whether again unicellular, multicellular, prokaryote, eukaryote, all those things will be considered in cell structure. Body organization. Body organization, sometimes the question is framed, they don't say it body organization, they say thallus organization. Okay, pay attention to that, just they are just twisting the words, meaning is same, body organization or thallus organization. So cell structure, body organization, and third one is mode of nutrition, saprophytic, that means living on dead, autotrophic, chemotropic, and all. Okay, we are going to study that. Next, mode of nutrition is there. Next is what? Mode of nutrition. Again, reproduction. Finally, sexual, asexual, with flagella, without flagella, and all those terms. And finally, they have done very important thing the R.H. Whittaker has considered is phylogenetic and evolutionary relationship. Okay, so how they are related to their ancestor and further generation, that is descendant. So this phylogenetic or evolutionary relationship they studied, these five criteria is often asked in a MCQ. Very IMP, very important. They will ask you this five criteria for five kingdom classification, 100%. Next thing they did was to put all prokaryotes in Monera. All prokaryotes. If you don't have nuclear membrane, put that organism into Monera. Next, all unicellular eukaryotes are placed in Protista. Most important. Okay. So these two kingdoms addition solved most of the problem. So if you go with this, again, prokaryotes Monera, and in Protista, they have gone with the they put, new thing has happened. Earlier, we have seen Chlamydomonas chlorella or diatoms, or these diatoms and dinoflagellates. They were placed separately with protozoans because of the cell wall. Here, multiple things are considered, and Protista, Chlamydomonas with chlorella, and Paramecium and amoeba, these two lack cell wall, these two have cell wall, but they are still put together in Protista. They are still put together in Protista. Correct. Pay attention. Chlamydomonas, chlorella, cell wall, without cell wall, still they are put in Protista. Why? Okay, earlier they were separated, but now they are put together in protista. Why the classification of organism changed? So this is one of the question which can be asked as assertion reason. So simply you can say this happened because criteria for classification was changed. In two kingdom classification, simple criteria was there that cell wall present or absent, present, plant kingdom absent, animal kingdom very oversimplistic and error prone. What has happened here? Pay attention. They have not gone with that. They have gone with the comprehensive criteria. This criteria is more scientifically accurate. So it is producing more scientifically accurate classification. Okay. So Chlamydomonas chlorella have proteins and glycoproteins in their cell wall, and these amoeba and chlamydomonas are both placed together in protista. Why? This happened because criteria for classification change. Assertion. This could be a reason and this could be a assertion. Next, what are the drawbacks of five kingdom classification? Viruses, viroid, prions. Okay, viruses, viroid, prion, and lichens are not included here, are not given any place in five kingdom classification. Okay, so why this happen? Because these do not fit normally. So what is a virus? Virus is a protein plus nucleic acid. We will come to that. So viroid again is a nucleic acid only. Prion again is a protein, and this is a combined organism like in algae plus fungi. So Whittaker could not place them anywhere in its classification. Next, this is the chart given in NCERT. So very important. Next, Monera. So cell type is prokaryotic, cell wall, non-cellulosic, polysaccharide and amino acid. So what is now this non-cellulosic, polysaccharide and amino acid? Okay, so it will come further in the chapter, but still here non-cellulosic means non-cellulosic polysaccharide here is glycan. Okay, and the amino acid is called as peptido. So the bacteria will have peptidoglycan cell wall. Bacteria will have peptidoglycan cell wall. For eubacteria, for archaebacteria, the composition can be different. So altogether considering these properties, they have said simply non-cellulosic polysaccharide and amino acid. Next, nuclear membrane. It is absent. Nuclear membrane. It is absent. Again, Monera. Body organization, one cell, cellular. Again, mode of nutrition, most nutritionally diverse group. It has so many types, that's such as in autotrophic it has two, chemoautotrop and photoautotrophs. In heterotropy it is also having two, saprophytic and parasitic. Next, again protista eukaryotic, cell wall is present in some, absent in some. Nuclear membrane is present, that's why it is eukaryotic. These 2 points are same. Mode of nutrition, autotrophic and heterotrophic. Simple. Fungi. Fungi again. Fungi is eukaryotic. Fungi again is eukaryotic. Come on. And cell wall, what is mixed there? Instead of cellulose, what is present there? Chitin. Nuclear membrane present. Complexity is multicellular or loose tissue. Multicellular or loose tissue. Okay. So from cellular we went to multicellular and the tissue is not compact but loose. These are completely heterotrophic, saprophytic and parasitic. Some one more mode is there when, okay, Next, plantae. Plantae are eukaryotic. Their cell wall is present. Cell wall is cellulose, and nuclear membrane is present. Body organization, tissue organization is present, and mode of nutrition is autotrophic, that is photosynthetic. That is photosynthetic. Next is kingdom Animalia. Cell is again eukaryotic. Cell wall is absent, characteristics of animal. Again, nucleus is present. Here in plants you found tissue and organ level. Here tissue, organ and organ system level, like digestive system and all. And finally they are heterotrophic, that is holozoic. Holozoic means they will eat either the complete organism or portion of organism as a bolus. And saprophytic, again dead and decaying things. So this is how we have completed 5 kingdom classification system. Next one is 6 kingdom classification by Carl Woese. Okay, so what is happening in 6 kingdom classification? That this is the most advanced type of classification. It was given by Carl Woese, and the criteria for this is RNA sequence. Criteria for this is RNA sequence. If anybody asks what kind of criteria is used for 6 kingdom classification, is RNA sequence, and that RNA sequence generally it is for rRNA. Okay, so rRNA sequence is used here. rRNA is present in ribosome. Ribosomes are factories of protein, which we will learn about in cell biology, or you have learned about this in 10th and 9th. So rRNA are present in ribosomes. They have taken the ribosomes and sequenced it. Their sequence, and where the more matching of the sequence, the organisms are placed together in. Where the sequences are different, they are placed in different group. Okay. Next, living organism, rRNA sequence and criteria for 6 kingdom classification. 3 domains. So we have 3 domains here and 6 kingdoms. So pay attention. First is going to be bacteria. First domain Carl Woese give is bacteria. Second is archaea, and third is eukarya. So earlier we had 5 kingdoms. So out of those 5 kingdoms, he just split Monera. If you want to pay attention, if you are paying attention, he just split Monera, and that Monera became the domain that is called as bacteria. Again, here eubacteria and archaebacteria are part of Monera. Same way they are part of this bacteria domain. Next is archaea. So archaea, in this archaea has only one kingdom. This domain, bacteria domain, has only one domain. Archaea domain has only one kingdom. Same way bacteria domain has only one kingdom, that is eubacteria. Okay. So what is happening here? Pay attention. Cell wall, the peptidoglycan is absent. In case of archaebacteria, the cell wall peptidoglycan is absent. But in case of eubacteria, U is true. U means true. Okay. So eubacteria, peptidoglycan is present. Peptidoglycan is present. Okay. So membrane is phospholipid bilayer with proteins and all. Okay, these many of the things, the terms are coming here. You will learn those about in cell biology, cell unit of life. Okay, so now just bear with it. So next is archaebacteria. Archaebacteria, peptidoglycan is absent. Some other proteins are present, and branch chain are present between two membranes. So even though its composition is bilayer, it becomes monolayer. Okay, so this is about archaebacteria. So this is a one kingdom eubacteria, archaebacteria. This is a one domain bacteria and archaebacteria. They have put all other eukaryotic animals in third domain, that is kingdom Eukarya, Protista, Plantae, Fungi and Animalia. Fungi and Animalia. Okay, so here we have completed the classification which is present in biological classification. Next, we are going to deal with the bacteria, kingdom Monera and all. One by one, we will go through it. I think, yes. So, here we have completed the classification up to 6 kingdom classification. Next video, we will going to complete the kingdom Monera, kingdom Protista, kingdom Fungi, introductory part of kingdom Plantae and kingdom Animalia, because they are separate chapters. So this lecture ends here. We have done up to classification. Okay, so up to classification we have covered. Next thing we are going to cover from kingdom Monera. We will complete in next lecture kingdom Monera. Thank you.